Hello students, in this video we will be discussing about the cell, the unit of life. Cell is the functional and structural unit of all living organisms. There are unicellular organisms and multicellular organisms. A single cell is capable of independent existence and it performs the essential functions of life. All cells have certain structures in common to carry out basic life processes. But differential distribution of organelles gives cells distinct characteristics. In living organisms, the metabolic reactions occur only in a delicately balanced environment that is different from any environment found in the non living objects. So, cells are the life supporting chambers which have such a special environment. A living cell keeps its chemical composition stable within its boundary. The term cell was introduced by an English scientist, Robert Hooke, in 1965. He examined thin slices of cork under the primitive kind of microscope which was available with him, and he noticed small boxes or compartments in the cork slice. These compartments were later named as the cells. These are the cells, the compartment like structures. Then later on, Antony von Leeuwenhoek first saw and described a live cell. Then Robert Brown later discovered the nucleus. The invention of the microscope and the later advanced technologies finally led to the invention of electron microscope. So the electron microscope revealed the detailed structure of the cell. Then comes the cell theory. Further studies with the help of electron microscope led to the cell theory, which was formulated by Schleiden and Schwann, and the cell theory was modified by Rudolf Virchow. This Matthias Schleiden, in 1838, he examined a large number of plants and discovered that the plant body is composed of plant cells. And same time, Theodore Schwann, another British zoologist, he studied the types of cells in the animals, and he found that the animal body is also made of cells. And these cells, they do have an outer boundary which is called as the plasma membrane. Now, based on these studies, the plant cells and the animal cells, they do have the similar characteristics, structural and functional similarities. So, Schleiden and Schwann together formulated the cell theory. The cell theory, which states all living organisms are composed of cells and products of cells. Then comes Rudolf Virchow in 1855. He explained that the cells divide and new cells are formed. So new cells arise from the pre-existing cells. So thereby the theories of Schleiden and Schwann and Rudolf Virchow together put the present day cell theory. So the cell theory is understood now as all living organisms are composed of cells and products of cells, and all living cells arise from pre existing cells. The cells are the basic structural units of living organisms, and the cells, they have cell wall in case of plant cells, and the cell wall is absent in animal cells, but both of them have a plasma membrane. So inside the plasma membrane, what you have is the protoplasm, which consists of the nucleus and the cytoplasm. And the cytoplasm contains or may contain various cellular organelles which are membrane bound in case of higher organisms like the eukaryotes, but in prokaryotes you will not find the membrane bound cellular organelles. This difference is there in case of the unicellular or the prokaryotes and the eukaryotes. The number of cells in organisms, in living organisms, plants and animals vary. Number, shape, size, all these in these organisms vary. Many cells are there in case of multicellular organisms, and unicellular organisms like amoeba, paramecium and all, they have only one cell, and the single cell performs the function of the whole body, the whole organism. Now the shapes of the cells also vary. Different shapes are there. Some of them are spherical, round, elongated, branched. So different shapes also there in various cells. Mycoplasmas, the smallest cells, they are too small, 0.3 micrometers in length, but the bacteria, they could be 3 to 5 micrometers. The largest isolated single cell is the egg cell of the ostrich. Then human red blood cells are about 7.7 micrometers in diameter. Nerve cells are the longest cells, and the cells, they vary in size and shape and functioning also, depending on the place where they are present and what function they perform. And the cells which contain the nucleus, and the nucleus carries or nucleus contains the genetic material, that is the DNA, and the other cellular organelles like the mitochondria, Golgi bodies, endoplasmic reticulum, ribosomes, and all these kind of cellular organelles which are membrane bound are present in the inside the cell in case of eukaryotic cells. Now we will see how the prokaryotic cells are. They are the single celled organisms which do not have a proper nucleus. They are represented by bacteria, blue-green algae, mycoplasma, pleuropneumonia like organisms. They are generally small and multiply very rapidly than the eukaryotic cells, and they may vary their shape and size, and examples are the bacteria, and for basic shapes of bacteria are the bacillus, coccus, vibrio, and the spirula. The organization of the prokaryotic cell is fundamentally similar, though they exhibit a wide variety of shapes and functions. They have a cell wall, which is surrounding the cell membrane, and the fluid matrix which is filling the cell is actually the cytoplasm, and there is no well defined nucleus. The genetic material is not enveloped by the nuclear membrane, and their addition to the DNA, sometimes circular DNA or single DNA is present. They may have small circular DNA which is outside the genomic DNA, so which are called as the plasmids. Now, organelles are found in these prokaryotic cells, like what is there in the eukaryotic cells, and they have something unique in the form of inclusions. These are the specialized, differentiated form of cell membrane, which are called as the mesosomes. That is the characteristics of the prokaryotes. These are actually infoldings of the cell membrane. Uh, the bacterial cells, they have a chemically complex cell envelope, which is consisting of a tightly bound three-layered structure. The outermost one is the glycocalyx, then the inner one is the cell wall, and then the plasma membrane. Each layer of the envelope perform a distinct function. They act together as a single protective unit. The glycocalyx differs in composition and thickness among different bacteria. Sometimes you may find a loose sheath which is called the slime layer in some of the bacteria, while in some bacteria the it may be thick and tough. Then it is called as the capsule. The cell wall determines the shape of the cell and provides the strong structural support to prevent the bacterium from collapsing or bursting. In bacteria also, the semi-permeable, the cell membrane or the plasma membrane is semi-permeable in nature. Then there are the special membranous structures, the mesosome, which is formed by, which are actually the extensions of the plasma membrane into the cell, and these mesosomes are in the form of vesicles, tubules, and lamellae. And these mesosomes, they help in the cell wall formation, replication of the DNA, and the distribution to the daughter cells. Mesosomes also help in respiration, secretion, and they increase the surface area of the plasma membrane, thereby increasing the enzymatic activity also. Then some bacterial cells may be motile or non-motile, depending on the presence of the flagella or the cilia. And the bacterial flagellum is composed of basically three parts: the filament, hook, and the basal body. And the flagella, the filament part of the flagellum is the longest portion, which extends from the cell surface. Besides flagella, the bacterial cells may have pili and fimbriae. These are also surface structures of the bacteria, but they are not helping in the motility or the movement. Pili are elongated tubular structures made of special protein, and the fimbriae are bristle-like fibers which are seen out on the surface of the cell, sprouting out of the cell. They are known to help attach the bacteria to the substrate. Then there are some ribosomes and inclusion bodies in the prokaryotic or the bacterial cells. The inclusion bodies actually they contain reserve materials in these prokaryotic cells. They are stored in the cytoplasm in the form of these vesicles. Phosphate granules, cyanophysin granules, glycogen granules, all these are stored in the vacuoles and they are the inclusion bodies in these prokaryotic cells. Then the ribosomes are associated with the plasma membrane of the cell, maybe about 15 nanometers to 20 nanometers in size. And these ribosomes are made of 50S and 30S subunits. So the prokaryotic ribosomes are 70S, and they are the sites of protein synthesis. Sometimes you will find many ribosomes attached to a single strand of messenger RNA, which is called as the polyribosomes or the polysomes. So these ribosomes or the polysome translate the messenger RNA into the proteins.